back to our class. Today we are going to be looking at life science grade 10. I received a lot of I've received a lot of requests. Just they're asking me to guide them using the distinction material for grade 10 to show them how can we, where do we, all those questions, find what you expect in the paper. First of all, I know that most of you have this so-called, if you don't have, you can download it from our website. Yes. This is what you expect to find in grade 10 exam. But now, when you come to what you call biosphere, when you come to what we call the biosphere, you have to talk about terrestrial aquatic biomes. In this regard, location of different biomes. So this is going to take you 8 marks. So basically, when you come to the distinction material, please make sure that this is what they are talking about. But this brings about the summary of everything. Biomes, check, look at the different colors. So follow that guide so that you are able to scoop out. These are the different biomes they are talking about. So please know at least the way how I've stipulated them there. But the most important thing is you need to know where is Savannah located on the map. At the Savannah, grassland, in Amakaru. We like to bring this map. So please make sure that you know how to find them in on the map. So based on this, because they're saying that location of different biomes in South Africa. So make sure you know that. When you go to the next point, I'm not going to use a lot of minutes, maybe 10 minutes will be maybe enough. So at least make sure that you are on the live. Then we have what you call the environmental environment and ecosystem. This one includes the energy flow diagram and nutrients. So here they talk about the concept of human activities and interactions, biotic factors and abiotic factors, physiographic factors. We like to ask this. Why? Because these ones, they confuse the students when they bring a slope like this, and then they ask you this side, the sun is this side, and they ask you what is being shown here aspect, because this is showing the direction of the sun, and then the slope, and then the altitude. So you need to know that. Let me show you what I'm saying here in the booklet. So these are the aquatic biomes. So when you come to environment, please know at least the definitions, because you can bring them in the terminologies. Biotic factors, these are factors which you are able to answer them very fast, and abiotic factors. But when you come to abiotic factors, we rarely ask about edaphic factors. When you bring about the edaphic factors, we bring about maybe amount of air in the soil in terms of experiment. But in most cases, I've seen many exams, you find physiographic factors. Yes, aspect, slope, and altitude. Please go when you know. This is what I'm trying to say. This, you see, we like to ask these questions, you see. So what is shown here? This, if here is the sun, so what is shown here? So it shows the aspect, and then the gradient is what called the slope, and how high this is. Yes, how high it is from the sea level is what you call the altitude. So you please go when you know those factors. We like to ask the physiographic factors in terms of the abiotic factors. And then when we come to wetlands, we ask about wetlands. When we ask about wetlands, at least go when you know 3 advantages of wetlands. what are some of the functions of wetlands? You know, stores water. We can get food from there. We can get papyrus. We can get grass for building the houses. Mud. We can get is home for many birds. So there are so many advantages there. So please at least you know that because it's also another aspect we usually ask in the what in the exam. So when we go to the next aspect, they're saying that water, light, yes, so we have talked about them. This one, trophic levels, producers, consumers, and decomposers. I know that you know this. This is very easy to answer. But what tricks the students is this? What tricks the students is this? What tricks the students is how energy is being lost. Calculate the amount of energy being lost. This. Why? From here to here, how much is 10% being transferred from one trophic level to another? Please know this, ne? This energy thing, ne? Yes. So please always use this distinction material book. You will be able to get it. You will be able to get the distinctions you are trying to look for. And then the different kinds of when you have something like this. Actually, this is a normal pyramid. Yes. And then anything which is starting with a small and then big and then small, something like that, we call it inverted pyramid. But this can not be for energy pyramid. On the pyramid which shows the energy flow is always like this. And in most cases, it shows few number of individuals, a lot of individuals, and then fewer individuals. Yes. So one tree, a lot of maybe butterflies, and then you have a bird feeding on the butterfly. You see, one tree, a lot of butterflies on the tree. There are a lot, and then there are fewer birds compared to the butterflies which are feeding on. So pyramid of number, pyramid of biomass can be like this, or can be like this, or it can be like this. Pyramid of number and pyramid of biomass can be like this. But pyramid of energy is always like this. Pyramid of number, pyramid of biomass, pyramid of energy, all of them can be like this. But only pyramid of energy cannot shift from this to this. But the other two can do that. So please don't go to the paper without knowing that. Sometimes we ask you this. What will happen to this if this is removed? What happened to this if that is removed and this is removed? So guys, try to read this. You'll be able to get. Trophic, this we like to bring this in the terminologies. Autotrophs, organisms which can produce their own food. Heterotrophs, those are organisms which feed on other organisms. Decomposers, organisms which break down the dead organic matter. Yes, like the saprophytes, the bacteria and stuff. So please go when you know. When you come back to the highlights here, I think we are done. Then we go to the flow charts. Here we are talking about nutrients, talking about water, meaning that the cycles. Basically, what we like to ask about the cycles is water cycle and nitrogen cycle. Even others, we ask them, but we like to ask what we call the water cycle and the nitrogen cycle. Yes, let me show you what I'm saying. So this is, I think I've explained this. But differentiate between a food web and a food chain. The arrow, what does the arrow indicate? All those things, just highlighting you because some students were asking me about this, the scope, and let me say, I'm saying the scope, or not saying that scope, I'm saying the prediction, and then where can we find in my book in the prediction. So in this case, today I've not used the questions because usually I use the questions and then I show you most likely questions to come and stuff like that. But now I'm just showing you where to read in the book. Yes, if you don't have it, just download it from our website, or you can just join our class and then choose on the, you will see there the button which shows join. Yes, and then you choose books, and then after that you can download the book. All right, then we go to Water cycle, yes, we like to ask this. We hide something here and then ask you what is that. I hide this and ask you what is that. So basically that's what you usually ask. And then we also ask, we like this, but don't usually come in the exams. We like the nitrogen cycle. Please, you can talk about others, but this, we like it too much. So make sure that at least you go to the paper when you have mastered this. Then we go to the next subtopic, that is classification. Here when you talk about classification, you have to know why do scientists use scientific names, why we say that just to prevent this confusion. The 5 kingdom system, so the different kinds of naming or classifying the organism. So please go when you know the 5 kingdom system, the 2 kingdom system, the 5 kingdom system, but we like the 5 kingdom system. And then we also bring these eukaryotes and prokaryotes. We said U means true nucleus. You can go to the videos I had already posted. You'll find them there. Yes, prokaryotes, eukaryotes. Pro means before. This means true nucleus, meaning that they have a true nucleus. The nucleus is the nucleus is bounded by the membrane. So we saw this one, the autotrophs, multicellular. So the 5 kingdom, yes, this is the 5 kingdom I'm talking about. Then taxonomy. Please, no. These systems, these systems created by these guys, which called a binomial, of which we use when you're talking about the binomial system. We use the genus, and then the species name. Don't forget that the genus, when they ask you to write something in the scientific name, the genus is always starting with a capital letter. For example, if I'm M.Saidi, yes, all right, yes, if that's it. I know that most of you have been knowing me as M.Saidi. Anyway, that's not a big deal. If I'm M.Saidi, yes, and ask you to write this in a scientific name, yes, so what am I going to do? I'm going to say Mosa is going to be capital S, capital M, and then side is going to be small s. So first letter is capital, Homo sapien, Homo with a capital letter, and then sapien with a small letter. I think I'm clear there. So we call it a binomial naming system or nomenclature. Nomenclature comes from word naming. Anyway, I'm not teaching. Then we go to, when we come back here, I think you can get those things, what I talked about. Prokaryotes, eukaryotes, I talked about. Differences between the two. This one has the true nucleus, has no true nucleus. So you can go and then read about those differences. History of discovery, history of life. Here we are talking about the geological time scale. In most cases, this you have to know how to use the geological time scale. This is what I'm talking about. Anyway, but you just need to know also the events which took place there. But you have to know how to use the geological time scale. I know that you have to be knowing by this time because even they brought it in the SBA. Yes, so they say that they are coming back. Please don't go back. Don't go to the exam without knowing the mass extinctions, the 5 mass extinction. Yes, please go when you know them. What is gonna cause is even the next mass extinction. Yes, and then the formation of fossil. Please go when you know how to explain the formation of the fossils. Fossil formation. If you come here in our book, you'll find it here. The fossil formation. The fossil formation is here. The 6 mass extinction, we talked about it. Fossil formation. Don't go without knowing dead, deposition, and then permeabilization, and then erosion, or sometimes called exposure or removal of the covering, and then the fossil can be exposed outside. So please go when you know these things. Don't forget that the paper is 150 marks. If it's 150 marks, then automatically you have to be, we have limited content. It means that we're gonna be testing almost everything. So please make sure that you, at least you go when you know this. As they said it, formation. radioactive, radiometric dating, and then carbon relative dating. Here we are using layers of soils. We don't know the actual age. While here we can know, yes, you date using the radioactive substance. Anyway, we go, here is the things we are talking about, different fossils. Please know how to these fossils, so methods of dating. And then when we go to the next is molecules, organic molecules. Here they talked about please know these carbohydrates. These ones we usually bring them in these ones, nucleic acid. So we can't bring all of them. Vitamins we usually bring them in objectives, but this or a passage. But carbohydrates, lipids, and the proteins, hey, those ones, yeah, please don't go without knowing those carbohydrates, the advantages, the advantages of, or the functions of this. Please make sure, don't make a mistake without knowing these carbohydrates, these carbohydrates. Then another thing which I should emphasize on is the food testing. When you talk about food testing, so after reading these ones, these compounds, you have to know how to test them. How do you recognize them? And then how do you recognize them? We said proteins, what do you use? Use burette reagent. And then starch we use iodine solution, blue black. Then this one, it turns from blue to purple if you use the burette test. And then how do we test for lipids? You can use the transparent paper, or you can use the two layers, the miscibility, whether they can mix or they cannot mix. Then glucose, let me talk about the glucose. Glucose, what do you use? We use Benedict solution. It's blue also, but it turns from blue to green to yellow to orange depending on the concentration. Yes, blue to green to orange to red. Sometimes they say red, sometimes they say brown. Yes, depending. And sometimes they used to say brick. No, not brick. Yes, brick red. When the brick is red, they call it brick red. But which color is that? Is red. So it's better to say red. So the types of carbohydrates, the monomers of carbohydrates are glucose and fructose. When we talk about the disaccharides, made up of two sugars, polysaccharides made up of many, sucrose and cellulose, glycogen in animals. Yeah, so please know that when it comes to the enzymes, please don't go to the paper without knowing these factors affecting the enzymes or enzyme activity. Temperature, yes. pH, yes. You have to know these graphs. So this is what I'm saying. So we have talked about this. Yes, we have talked about this. So please, when you go to the enzyme part, go when you know these graphs. Yeah, this mechanism, key and lock. We also I think when I was teaching, I was very clear about it, that don't go without knowing the key and lock hypothesis. How does it how do the enzymes move? Yeah, these graphs. Don't go without knowing these graphs. You see those graphs. And then lastly is the cell, the structure of the what? The structure of the cell. Yeah, this one is a little bit scooped out. The structure of the cell. Yes. Plant cell and animal cell. We can bring a plant cell or we can bring animal cell. Or sometimes we can bring one part of animal cell and one part of plant cell. So when they say it here that you must know the structure of makeup of the cell, proteins, so we have done with that. Make sure that you know the size, shape of all these organelles, meaning that you have to know the plant and animal cell. So when you come here, please know how to draw the plant, how to label the plant and animal cell. But I usually tell my students to draw so that they master what they are labeling. So the difference between plant cell and animal cell, please know that. And then these organelles, the organelles, obviously we might not bring the cell membrane. If we bring the cell membrane, we can ask. But what we usually bring is either a mitochondrion or a chloroplast. You can't go to the paper without knowing a nucleus, nucleus. You remember when I was teaching you, I say that nucleus you have to do with your what? Your hands. And then the chloroplast, you cannot go to the paper without knowing the structure of the chloroplast, and you cannot go to the paper without knowing the structure of the mitochondrion. So the mitochondrion, the chloroplast, and the nucleus, please go when you know them. Even here, I think they highlighted them. I saw them somewhere. They say that you must know. Yes, they are here. Drawing of at least one: nucleus, mitochondrion, and chloroplast. Please make sure that you know this. And then lastly is the bar graph, line graph. Make sure it has a title. Make sure that if it's a bar graph, these bars have the same size, same space. Make sure that yes, everything there is what you have written here, the x, the y-axis, the x-axis, the title. This one must have units. Even this one must have units. No units here. So make sure that if it is a pie chart, convert it into degrees. And then if it's a line graph, make sure that there is no line graph which looks like that. There is no line graph which looks like that. But there is a line graph which can be like that, and there is a line graph which look like that on the scale. So make sure that you know those points. I think that is it. I said that I'm not going to waste too much time here, but I beg you that please and please don't go to the paper without knowing this nucleus, mitochondrion, and then the chloroplast. I think that's it, and that's the distinction material. If you want it, make sure that you download it and then use it. That's it. Yeah. So then meaning that the next term will be talking about the cell cycle or cell division. So that's it. God bless you. See you again. I'll try to discuss the paper maybe immediately after you have written. M.Saidi as usual.